Most people think of water as something to drink, to irrigate farmland, or to generate power. Few would ever connect a glass of water to a vial of cell therapy and production. But today, that connection is growing closer than ever. A recent United Nations report warns that the world has moved from a water crisis into water bankruptcy. Nearly 3/4 of the global population now live in water-insecure countries, and about 4 billion people face severe water scarcity for at least 1 month every year. For the biotech industry, this is not a distant environmental issue. A joint analysis by WWF and AstraZeneca found that over 70% of pharmaceutical manufacturing sites worldwide face high or extremely high water quality risk. Water is shifting from a resource problem into an industrial variable. What's truly fascinating is that several frontier breakthroughs in water technology happen to find their exact counterparts in life science. First, microbubble technology, generating micron and even nanoscale bubbles in water to dramatically boost oxygen transfer efficiency. Originally used to aerate industrial and agricultural water, this technology has now entered stem cell culture. A 2026 study found that nanobubble oxygenation reduced oxidative stress markers in hypoxic stem cells by roughly 20%. A similar principle is being applied in next-generation wound dressings, where sustained oxygen release promotes tissue repair and new blood vessel growth. Second, electrolyzed water and the molecular hydrogen it contains. Scientists have clarified that the true active ingredient isn't some mysterious change in water structure, but measurable dissolved molecular hydrogen, which selectively scavenges cytotoxic free radicals. Multiple human trials have shown that hydrogen-rich water can lower inflammation markers and improve mitochondrial function, even improving coenzyme Q10 levels in patients with non-alcoholic fatty liver disease. Third, the effect of far infrared and terahertz frequencies on water molecules. Rigorous physics experiments confirm that specific frequencies do produce real resonant interactions with water's hydrogen bond network, but only on a femtosecond timescale. Mainstream science also cautions that this is a fleeting physical phenomenon, and extrapolating it into long-term activation with specific biological effects still lacks reproducible evidence. That honest restraint is itself part of what makes water technology rigorous. Fourth, the extreme water purity demands of organ-on-chip systems. Organ-on-chip pioneer Donald Ingber has pointed out that even minor ionic fluctuations in culture media can distort drug testing results, which is exactly why pharmacopoeias worldwide demand near uncompromising purity standards for water for injection. The credibility of an experiment hinges on whether that single drop of water is pure enough. Together, these four threads offer a simple but profound insight. Every advance in water technology is ultimately measured by whether it can be genuinely sensed and used by living systems, oxygen transfer efficiency, molecular hydrogen concentration, hydrogen bond vibration frequency, or the purity grade of a single drop of water. This also reminds us that what makes a water technology trustworthy isn't how compelling its story sounds, but whether it can be repeatedly verified through peer-reviewed experiments and backed by measurable data. Biotech's next breakthrough may not arrive in some isolated flash of insight in a lab. It may be hiding in these seemingly basic details of water itself, details that decide everything. Whoever can make water more precise, more verifiable, will be one step closer to the next stage of life science.